Hello everyone. Well, today you're at home in the comfort of your home and probably on your favorite chair. Hopefully you have some chips with you to be entertained. And well, unfortunately, we've got to do this lecture, which is actually meant to be a time for your, it's scheduled in your timetable. So I hope that you enjoy this session and you'll be able to gain as much as you can. So in case you do not know me, I'm Mr. Rashid. I'm one of the team members in the microbiology lecture team. And well, you look at the big picture about why we're having our session as BCR is meant to be a situation where if let's say there is some outbreak of a disease or infection or any kind of uh, casualty or calamity, so lessons should still be able to continue. So we mentioned, I mentioned just now about the condition where you may have an outbreak of an infection such that it's better for you to be at home. Lesson can still go on, right? So picture a scenario right now where there is an outbreak of a disease. So there's a possibility that you may get infected. So if you do get infected, that means that you are a susceptible host because you have become part of the in what you call this thing, the chain of infection. So if let's say you do become a susceptible host, you fall ill, you are going to need some form of treatment and therapy. Very often, left on your own without any kind of antimicrobial treatment, your body will hopefully be able to combat the infection and be able to recover. But very often, it takes a very long time to rely on your immune system to combat the infection. So as a result, there is therapy, and that therapy is actually a form of antimicrobial therapy, and very often we call them antibiotics. This is something that you're very familiar with. You probably have had it before. Okay, so keep that in mind as we are going through this lecture. So very often you pop in this medication called antibiotics. What are you doing? What is this medication doing in your body? How does it help you? How does it combat those pathogens and those bacteria and things like that? Okay, let's break down in greater detail now, step by step, what is an antibiotic. Okay, so there's a couple of things that we need to cover with regards to the objective of this lecture. We need you to be able to understand what on earth is an antibiotic. Okay, with regards to MIC, for this series of lectures, we are focusing mainly on antibacterial forms of antibiotics, because there are other forms of antibiotics that can combat protozoa, fungus, and fungi, yeah? and also sometimes even viruses to inactivate them and things like that. But for the purpose of our lectures, in terms of our curriculum, we're going to focus mainly on those which are antibacterial. Okay, so that's point number 2. Point number 3, we're going to go on with the modes of action, or another way of describing it would be how it actually works. Okay, what does it do in terms of combating the pathogens? Okay, so antibiotics, there are 5 main ways in which they can kill or stop the growth of bacteria. So we'll go through that in greater detail right now. Oopsie. All right, so now when we talk about antimicrobial, we are looking at substances which are like antibiotics. These antibiotics are usually chemical substances, and these chemical substances, they are usually obtained either synthetically, that means they can be made from the laboratory, or some kind of from the industry or factory, like in the pharmaceutical company, like Schering-Plough, or maybe GlaxoSmithKline. You got Merck Sharp and Dohme. These are big companies that make antibiotics and drugs in general. So they can make them from all these factories or industrial from this tuas and things like that. Well, very often in nature, there are substances that are produced by bacteria as well as fungi. And this is what we're going to look at. These bacteria and fungi, they actually release substances from them. And these substances have been discovered by this scientist called Alexander Fleming. In the process of his research, he observed that when plates of bacteria have got contamination with fungus, this fungus was, it started out with penicillium. This is a fungus. On the plates, he discovered that when these bacteria have got this contamination of this penicillium, where this penicillium had been, bacteria don't seem to be able to grow. So that indicated to him that, aha, this penicillium fungus actually released a particular substance that seems to be able to control or kill this bacteria. And hence came about the discovery of antibiotics. And what was the first antibiotic to be discovered? The penicillin. And penicillin was obtained from the fungus Penicillium. Okay, all right, so let's continue from there. So, you know the antibiotics, they have an effect on bacteria, fungi, viruses, and protozoa. So when you say it has an effect on, say, virus, be very careful with that, because viruses non-living things, so you cannot say, aha, this antibiotic killed the virus, because viruses were never alive in the first place. So you must always say inactivate. Okay, so instead of killing the virus, you must say it is inactivating the virus. Okay, so keep that point in mind. Now, other things you need to take note with regards to antimicrobial therapy is that when you want to kill or stop the bacteria from growing inside your patient, you do not want the drug to also kill the patient. Imagine giving an antibiotic that kills the bacteria and kills the patient as well. What kind of a drug is that? You're going to become a murderer instead. Okay, so keep that point in mind as well when we talk about antimicrobial therapy, because this antibiotic must be able to selectively be toxic or have an effect on the pathogen, in this case the bacteria, but it is supposed to not have any effect on the person who take the drug. Okay, so keep that point in mind. Important, all right? Okay, next slide. All right, now antibiotic action. There are two ways in which this antibiotic can affect the bacteria. One of them is by being bactericidal. Do you see that? The word bactericidal? Oh my God, the way I'm controlling this mouse, it's horrible. So bactericidal means that it kills the bacteria. Okay, if let's say an antibiotic can kill fungus, it will be fungicidal. So if let's say in this situation, this antibiotic kills the bacteria. So this antibiotic is a bactericidal antibiotic. Can you remember that? All right. So on the other hand, there is another way in which antibiotics can affect the bacteria. It can stop the bacteria from growing. If it stops the bacteria from growing, it means that it's bacteriostatic. Remember, huh? Okay, so there's a difference on the effect. If it kills the bacteria, it's bactericidal. If it stops the bacteria from growing, it is bacteriostatic. Can you remember that? Okay, remember, important, all right, now the other thing you need to take note about bacteriostatic is this. You might think that it stops the bacteria from growing, so that means the bacteria is still going to go on living forever. Am I right? Like with all things in life, all living things have a lifespan, so the bacteria will eventually die. So if it stopped from growing, it's unable to perform binary fission, so it will not keep on growing and infect the patient, right or not? So eventually the bacteria will die, so the patient will slowly recover as well. But if you were to think about the effect of antibiotic, which one do you think would be a better antibiotic? The one that's bactericidal or bacteriostatic? Think about it. We're going to come to that very soon. All right, so remember that. So in this case, if it's bacteriostatic, it has an inhibitory effect. Whereas if it's going to kill the bacteria will die. Okay, all right. Next slide. Okay, now these are the desired characteristics of an antibiotic. Okay, what will that be? It has to be selectively toxic. Remember what I told you just now? You want to be able to affect the growth or kill the bacteria. You do not want to kill the patient. That will be xiao. Okay, all right. All right. Now, you prefer an antibiotic or an antimicrobial drug that is bactericidal compared to bacteriostatic. That is the one I would say to think about. Now, okay, bactericidal is a better antibiotic drug. Now, you want something that can be delivered to where the infection is. If let's say if it's on the skin, you would like to have an antibiotic that can be topical. That means you can apply it onto the skin where it will be able to affect only that region where the infection is. You do not want to take something that would go into your bloodstream, go all over your body and all that. So it's just too massive. Okay, so that would be a preferred one. You prefer something where it can be directed towards where the site of the infection is. Okay, so that's the third bullet point. Okay, third bullet point. Am I getting better at my control? Not too bad, quite koya. Okay, all right. So you would prefer an antibiotic or an antimicrobial drug that does not cause allergy. So something that is non-allergenic would be best. Can you imagine somebody who's taking antibiotic and then his face completely gets swollen, he starts to tear, and that can result in what is known as an anaphylactic shock, and the patient can die from an anaphylactic shock instead of the microbial infection. Can you believe it? It's crazy, isn't it? So that's why it's important to be very careful about the prescription of drugs. So doctors have got a huge weight on their shoulder to make sure they give the correct drug for patients. Okay, so of course you want something that is reasonably priced. You do not want something that costs a few thousand for just one tablet. That would be crazy. Okay, the patient will die from hunger because there's no food to buy, no more money already. Okay, crazy. All right. So remember, in exams, I always see students who say, "Oh, you prefer a cheap antibiotic." Now, the word cheap is usually avoided in academia as well as in exam papers and all that, because that's what we talk about when we want to buy handbag. Oh, cheap handbag, cheap perfume, and things like that. Okay, but for drugs and things related to science, you say reasonably priced or affordable. Those are good words to use. Okay, next slide. Now, when we talk about antibiotics, there is yet another way to group antibiotics. Earlier on, we talked about antibiotics that are bactericidal, and there are antibiotics that are bacteriostatic. This is another way to classify antibiotics, whether they kill both gram-positive bacteria as well as gram-negative bacteria. If they have this effect, of killing or stopping growth of both gram-positive as well as gram-negative bacteria, that means that antibiotic is a broad-spectrum antibiotic. Okay, now if on the other hand it has this effect of killing or stopping growth of only gram-negative or gram-positive only, just one type, that will be a narrow spectrum. Not so fantastic. You prefer one that is broad spectrum. Naturally, right? Okay, all right. All right, now these are the 5 important modes of action, or what's written in the slide here. It says mechanisms of antibiotic action. So remember, if in the exam you see mechanism of action or mode of action, they are talking about the same thing. Okay. So there are 5 modes of antibiotic action. The first one is to inhibit cell wall synthesis, and you can see the word inhibition is used in 4 of these 5 points. So the only one that is different is point number 4, which is disruption of cell membrane. So this will help you to remember all of them. You can just remember almost every one of them is inhibition of something. Inhibition of cell wall synthesis, nucleic acid synthesis, protein synthesis, or metabolic process. This is one way to help you remember these 5 modes of action. And the 5th one is to disrupt the cell membrane permeability. Okay, this you need to remember a little bit about cell membrane function. We'll talk about that very soon. Now, what else you need to know with regards to all these 5 actions? You need to go back to try to remember cell function. The parts of a cell, you need to remember in your first lecture you learned about prokaryotic cell as well as a eukaryotic cell. So I want you to try to recollect all these things all over again and try to remember the structures within these cells as much as you can. We will cover them in great detail in the next semester under the module called Cell and Molecular Biology. But for now, try to remember at least cell membrane, ribosome, also try to remember things like your cytoplasm and all that. So those are important structures that you need to remember that distinguishes between your prokaryotic cell as well as your eukaryotic cell. Because we are focusing on the effect of antibiotics on bacteria, so at least try to remember all these things about the cell wall of the bacteria, the nucleic acid of the bacteria. And nucleic acid, please try to remember, if you have your notes with you, write down in your notes right now. Nucleic acid here refers to DNA and RNA. Are you writing it down now? I'm watching you. Write it down now. Scared already, yeah? Okay, so remember, nucleic acid, please write down. Nucleic acid is an umbrella term for two types. What are they? One is your DNA, the other one is your RNA. We're going to talk about them quite a lot later on, okay? And then we're going to talk about protein synthesis as well as metabolic function. What on earth is metabolic process? We will go into detail very soon. So remember, there are 5 modes of actions. So let's go. This is a snapshot of where your antibiotic can have an effect on. Okay, the first one, I'm pointing at this one right now. Okay, inhibition of cell wall synthesis. That means it affects the cell wall. Second one, protein synthesis. As you can see here, down here, it affects the membrane. This will be the disruption, the odd one out, the one that's not inhibition. And then this one is inhibition, but for metabolic processes. And this one is inhibition of nucleic acid synthesis. So you can see the words here, DNA, which is DNA, and your RNA, which is your RNA. Yeah? Okay, all right, please don't say it the way I just did. It's DNA and RNA. I was just kidding. All right. Okay, let's go on now. So first point, inhibition of cell wall synthesis. That means it inhibits the cell wall making. So the bacteria is unable to make cell wall around itself. So if the bacteria is unable to make the cell wall function is no longer available to the bacteria. What is the function of the cell wall? You don't know, right? That's why you want to study. Now go and remember. Cell wall provides shape and a certain degree of protection to the cell structures within the cell itself. Okay, so the main function is it gives shape. So if it doesn't make cell wall, the cell will not have the shape, the rod shape lah, the arrangement and things like that. Okay, so you need to understand that without the cell wall, it doesn't have the shape, so slowly substances can go in quite easily in the cell, and the cell will slowly lose its integrity, and eventually the cell will burst. Okay, so bursting is where you get the disruption of the cell. So you see this last bullet point, cell rupture means the cell lyses. The word lyse means to burst. Okay, kabong. All right, so you need to understand both bacteria, gram-positive and gram-negative bacteria, have got this structure or material called the peptidoglycan. So if it's a gram-positive bacteria, it has got a thick peptidoglycan layer. If it's a gram-negative bacteria, it also has a peptidoglycan layer, but it's a very thin one. Okay, so remember, thick, gram-positive, thin, gram-negative. Can you remember? All right. Now, you see what happens with this antibiotic is that it actually affects the cell wall synthesis. It does not allow the bacteria to be able to make the cell wall. So without the cell wall, it's got no protection. So substances will go into the cell. The cell will eventually start to lyse or rupture. Okay. So examples of antibiotics that have this effect will be your penicillin. Okay, the one that was discovered by Fleming, and cephalosporin. Okay, now these day and age, you hardly see people or doctors prescribing penicillin. Okay, it's mainly because this antibiotic has been rendered to be quite most bacteria are very resistant to this antibiotic, so it's kind of useless. But you will get to see and work with this antibiotic in one of your practicals called the Kirby-Bauer sensitivity experiment. Okay, you will see that later on, not so soon. All right, so it'll be good to remember at least one of these antibiotics, and these are actually able to inhibit cell wall synthesis. Okay. All right. Now, this is a diagram to show you that without the peptidoglycan, so the cell will lose its ability to have that shape. So eventually, substances will be able to get in. So it's very easy for the cell to pitia or lyse or kabong, explode. You can imagine, you know, while you're sitting on there, your body will start to move, as though there's a huge explosion inside your body. Huh? How exciting! Huh? You wish that would happen, right? No lah, of course not lah. Just kidding lah. Okay, Next slide. Oh. Now, this is your nucleic acid. So you need to understand. We have been talking about bacteria, how it affects the bacteria. This diagram that you see on the right, it says it is a eukaryotic cell. So this is useful for you to know anyway because our cells are like that. Don't forget, antibiotics can affect your eukaryotic cells as well. What kind of eukaryotic cells that you want the antibiotic to go against? Those pathogens which are of fungus or the protozoan types. So like your plasmodium, or let's say for example certain your candida for example, there are fungicidal antibiotics as well. So these kind of fungicidal antibiotics, they can affect your eukaryotic cell as well. Okay, so please understand that. Open your mind, don't just think about your bacteria, because your other pathogens which are of the other kingdom, those other types, they will have the eukaryotic cell, and there are antibiotics that can affect them as well. So when we talk about inhibition of nucleic acid synthesis, we are looking at affecting the DNA synthesis, looking at the RNA synthesis. So this particular first line that I've drawn for you, this DNA to DNA, what does that mean actually? It simply means that for the bacteria to be able to grow and perform binary fission, so that it can form from one parent cell split to form two cells, before it can do that, it must make a copy of its DNA so that two of the daughter cells, let me draw two daughter cells. This is the parent cell, split to form two cells. Two little baby cells there. Cute, huh? Oh, it looks like a man carrying something. Okay, so you get two daughter cell. One daughter cell here, one more daughter cell there. Okay, drawing terrible. Sorry lah, brothers and sisters there. Okay, so listen. Parent cell splits into two by binary fission for the bacteria. So you need to understand, before it can perform this, it must replicate its DNA inside, that circular DNA inside that cell. So to be able to replicate, it must use its DNA to make another set of DNA. That is what we call DNA replication. Can you remember? Remember, important, huh? Because in second semester, you will be learning all these things in your cell molecular biology. Okay. Can, huh? Okay. All right. So go on. So please understand that if you can inhibit this process of replication, that means this antibiotic has an inhibitory effect on the DNA synthesis process, which is the nucleic acid synthesis. Okay, all right. Now, this is a simplified diagram of what Dr. Alpana had put in the slide about the eukaryotic cell just now. Take note, DNA is required to make this molecule called RNA. RNA is required to make this molecule called protein. We have lots of proteins in our body, and all these proteins from your hair, your nail, your saliva, the mucin in your saliva, your tears, all those proteins that you get in your tears, all these proteins are synthesized in your body. How is it able to make those proteins? Because of the food that you consume, all those proteins that you consume, your chicken lah, your fish lah, your tofu, if you're a vegetarian, all these things get digested and broken down into amino acids. Am I right? So these amino acids will be absorbed. Remember, your AAP, you're learning all these things. So try to make connections right now. So all these amino acids will be used to make those proteins required by your body. So those amino acids are joined together and form this nice beautiful hair that you see on Mr. Rashid right now. Okay, so the same thing happens with your body as well. Your own body will make its own protein. Where does it get all these amino acids from? From the food you consume. Okay, so if you can remember that, you will also understand that your RNA is required to be able to put all the amino acids together to form the protein. Now, this is very clear. To be able to make DNA, to convert it to, or rather to use the DNA to make a molecule called RNA, that is your RNA synthesis. But what about this arrow, this blue arrow that you see down here that I'm highlighting for you right now? This blue arrow is actually to show your DNA, the bacteria. It uses its chromosomal DNA to make a copy, an exact copy of another DNA molecule, so that after the cell splits into two, each of the daughter cell will have one set of the chromosomal bacterial DNA. All right, so this blue arrow that I'm highlighting one more time down here for you is the replication process. Okay, whereas this one is the making of RNA process. This one is called translation, which is not under your nucleic acid synthesis. Please be very clear about this. This is not under this one. Got it? Okay, for this one, let me draw the third line for you. For this one, it is this and this. You are blocking either one of this. So we'll talk about this very soon. Okay? Now, if let's say an antibiotic can block this process, this is an example for your antibiotic that you have. Ciprofloxacin is an antibiotic. What it does is that it prevents or inhibits the DNA replication process. Okay, if it prevents the DNA replication process, that means it is blocking at this point. Okay, it blocks down here. On the other hand, rifampicin, for example, or rifampicin, this antibiotic, what it does is that it blocks the synthesis of the RNA. Please remember in your exam, never write that the DNA is converted to RNA. Wrong concept, sinful. Cannot go to heaven if you say that. Okay, you must say that DNA is required to make RNA. So to be able to do that, there are enzymes involved. Okay, every one of these processes, enzymes are involved. Okay, so what does this rifampicin or rifampicin do? What it actually does is that it prevents the enzyme from being able to use the DNA to make the RNA. All right, understand? So just take note of that. So this number one highlights the DNA replication process is being inhibited, whereas for number two, it means that the process of using DNA to make RNA is inhibited. Okay, so this collectively, both of these, what are their actions? Both of these, their actions will be to inhibit the nucleic acid synthesis. Can you remember? Okay. So far so good. Is it too intense? Are sparks starting to come up from your head? No, no smoke coming out yet? No? Okay, very good. So take it easy. All right, don't stress. Here comes the third point we're going to talk about, and this is inhibition of protein synthesis, not the nucleic acid synthesis. The diagram looks similar, the slides look similar, but this one focusing on point number 3. The earlier things that we were talking about is your nucleic acid synthesis, right? It affects replication of the DNA, and it affects the transcription from using DNA to make your RNA, correct? Now that is for your nucleic acid synthesis. But for this one, we are focusing only on the translation. Okay? All right, so what happens during translation? Well, you need to see. Protein synthesis involves the substance, this particular organelle called ribosomes. So some antibiotics can affect the ribosome to prevent the ribosome from making these proteins, from joining all these amino acids to make the proteins. Okay, so without these proteins, the cell cannot do its function, so eventually the cells will die. So the bacterial cells will die. Okay, so that's how it inhibits the protein synthesis. All right. Okay, it's true that proteins are a predominant organic molecule. So many of the cells, many of our cells, in fact almost all cells, the large content in our body, apart from water, is actually your proteins. And because many of these proteins are in the form of enzymes, they do a lot of functions. Sometimes they've got structural functions as well. So we will talk about that in greater detail, but keep this point in mind. No protein, very likely the cells are going to die. So that's how some of these antibiotics can kill your bacteria or stop the bacteria from growing. So keep that point in mind. All right? Okay. So remember, there are quite a number of antibiotics. You probably, some of you've probably have ever had eye infection or ear infection. Very often, the antibiotic that's given for such an infection will be chloramphenicol. Chloramphenicol is very interesting. This is one of those that can be used topically for the eye or the ear because it's not given in the body. Why? Because it has the ability to cause kidney damage. Your damage can be kidney. Your kidney can be damaged by this antibiotic. Why? So you remember what I told you just now about you want antibiotic that is selective. So you do not want something that can kill you. So that's the reason why chloramphenicol is suitable for topical application. Apply at where the infection is so that it doesn't get into the bloodstream. It doesn't go into the system. It doesn't go systemic. So if it goes systemic, it will actually cause damage to your kidney. So keep this kind of useful information in mind, because later on you'll be learning about pharmacology and you will probably be exposed to this kind of drugs. So whatever I'm telling you now will be useful, maybe not now but eventually. So this don't learn by just remembering things, but try to make associations by making connections. So later on when you learn about pharmacology, you remember, aha, during this PCR lecture, there was this half botak man whom I saw in the video. He was talking about chloramphenicol used as a topical application. So it rings a bell, so you learn. Okay, so all information that's given to you is never a waste if you treat it with the right attitude. All right, next slide. So in this case, you can see that photosynthesis, like for example, antibiotics like gentamicin, erythromycin, what does it do? It blocks terpene at the point of the translation. It blocks it so that your RNA is not being able to be read by your ribosomes to be able to join amino acids together to form your protein. Okay, can you remember? All right, so if you inhibit photosynthesis, that is an antibiotic that can also kill or inhibit the growth of your bacteria. All right, so remember, we are not just talking about bacteria, it can also be other pathogens as well. All right, now disruption of cell membrane, you need to remember what is the function of the cell membrane. Is to control the movement of substances into the cell and also what can come out. If anything and everything can go in and out, it will be like a shopping center, things can go in and go out. It cannot, it's not like that. The cell controls things that are supposed to be able to come in and not suka-suka come in at any time. All right, so please keep that point in mind. But there are antibiotics that can puncture lobangs, big holes into your cell membrane of the bacteria. So if you can puncture holes inside, that means all the cytoplasm will come out like the intestines when you try to disembowel a person. You just cut like you perform harakiri. You cut your abdomen. Don't get ideas about how to commit suicide, please. I'm just letting you know. The Japanese, when they want to commit suicide, they actually stab themselves with a sword. You know, right? So you can imagine all the intestine will start to spill out. Okay, so that's exactly what's going to happen to the cytoplasm. Okay, this sounds like a very, very graphic movie, very scary. So you get the idea what I'm trying to tell you. Okay, so remember, if there's a big lobang in the cell membrane, so you can imagine all the cytoplasm starting to leak out. So everything's going to come out. So what happens to the cell? It will surrender with a white flag, just like what the Japanese soldiers will do. Okay, they never surrender. They commit suicide instead. All right, so metabolic processes. What on earth is a metabolic process? I don't know. What am I doing down here if I do not know? Okay, so metabolic processes usually refer to very important processes that occur within the cell, and among them will be making ATP, for example. You know what's ATP, right? It's an energy molecule that your body needs to be able to stay alive. So just imagine 没有 ATP 咯. So what happens? So your bacteria will die. No ATP, no energy, the bacteria will 死了, mati, will jatuh poita. Okay, all right. So you get the idea. All right. So that is an example of a metabolic process. Well, there are many other metabolic processes, and very often your body usually tries to make folic acid. Folic acid is a very important molecule required to be able to make DNA and RNA. So imagine, no folic acid, your body doesn't synthesize folic acid, no DNA. No DNA means there will not be cell division to be able to make new cells. No new cells, that means you're not able to make more of your red blood cells, because your red blood cells have a certain lifespan. Eventually they will all die. So as a result, it will not be able to make more of your cells in your bone marrow to be able to make more erythrocytes and your leukocytes and things like that. So what happens to you? What happens? You're going to die. So imagine if, let's say, you can control so that these antibiotics only affect your pathogen and prevent the pathogen from being able to make more folic acid needed to make DNA and RNA, you are affecting the cell's metabolic processes. Okay, understand? So if you can stop the bacteria from performing metabolic processes, you are actually inhibiting it by making sure that it is not able to grow or making sure that it dies. Okay, if it dies, that means that antibiotic is, recall what we talked just now. It will be bactericidal. If it only makes the bacteria stop growing but it's still alive until it eventually dies, that type of action is bacteriostatic. See, I'm helping you remember. I'm helping you revise as well. See, I'm such a good teacher, right now. Okay, must carry yourself a bit lah for motivation. I mean, hello, it's already way past 6 p.m. right now. What am I doing down here? So I must motivate myself that way lah. Okay, got it. Let's go on. Well, and we are back after that very, very scary looking thing. I'm not so sure what happened. Ew, I still cannot move on. Why is this so? Okay, I'm gonna come out again. Sorry.